present in that. Okay, so that's why the all plants leaves are appear in just wait. Okay, now I will share that material. Okay, we can discuss about one by one. So what is that hemoglobin? Hemoglobin is one type of the pigment that is this is the structure of that hemoglobin. Okay, so that is contain the four firing ring and a globin chain. Four firing ring and a globin chain. Okay, so that four firing ring is this is a four firing ring. Four pyrroles are surrounded by the four corners in that ring. In middle of that occupy the what? Iron. And remain these are the globin chain. Different types of proteins are there. These are the globin chain. Globin chain, that is also different role is there. These are the globin chain and term. That is the protein. So many proteins are there. They surrounded with that. So what is the role of this hemoglobin? Is capture the this hemoglobin is a 5 6 coordinate generally. Then here what 5 coordinate is occupy. How to occupy? The four corners are occupy the four positions. One pyrrole, two pyrrole, three pyrrole, four pyrrole. And the fifth position is occupy the histidine molecule or histidine, one of the protein of that histidine. That is a proximal histidine on term. That what is that? Proximal histidine. And the sixth coordination sphere, iron, the sixth coordination sphere is slightly bind with the or loosely bind with the or van der Waals forces bind with the water molecule. That is not considered in coordination number. That is van der Waals forces are there. Okay, so in this is a hemoglobin structure. So what is the hemoglobin structure? Means heme is the four firing ring, globin is the protein. Okay, both are connect. That is forming. We can consider that what hemoglobin. So in that hemoglobin, two parts are there. One is a four firing ring, one is a globin chain. Okay, so in that four firing ring, in middle of that or center of that is iron is the element. What is that? Iron is the element that is four corners are occupied the four pyrroles below the or above the plane. Any one plane is occupied the one of the histidine molecule or histidine protein. Okay, that is a proximal histidine protein. Then sixth coordination sphere is loosely binded or van der Waals forcefully binded the water molecule. Okay, so this is the hemoglobin structure or this is a deoxyhemoglobin. We consider about the what? Deoxyhemoglobin. Okay, so this is occupy the uh, bind with the oxygen from the surrounding and convert the oxyhemoglobin. Convert the oxyhemoglobin means what? In this water molecule is in that water molecule is what bind with the oxygen. That is oxyhemoglobin. After deoxyhemoglobin is the 5 coordinate that is can bind with the or means approach with the oxygen from the surrounding of the oxygen is there no, so atmosphere that oxygen bind with the iron and that is convert the oxyhemoglobin. So in that oxyhemoglobin this is a globin chain is there no, this globin chain or the transport the oxygen from one place to various parts of the living being. Okay, so the O2 is required for the living being now. So that O2 is supply one place to another place means all parts of the living being or our human being body parts. Okay, so this is a globin chain is the role. Okay, that is the oxyhemoglobin. Deoxyhemoglobin is convert the oxyhemoglobin and transport the oxygen from one place to various parts of the body. Okay, so this is what is the role of that? The globin chain is the role of that. Oxygen is accept the iron is accept the oxygen, means 4 5 ring is accept the oxygen or capture the oxygen, and globin chain is transport the oxygen. That is role. Then we can show that also the deoxyhemoglobin also can show. So this is the hemoglobin, oxy and deoxyhemoglobin. Okay, so this is the deoxyhemoglobin. It's a deoxyhemoglobin. This is the oxyhemoglobin. So what is the spin of that? Spin means what is the nature of the iron in both. So here iron is contain the central atom that is present in Fe plus 2 state. What is that? Fe plus 2 state. In that Fe plus 2 state is contain how many electrons of the Fe plus 2 means what is the iron atomic number? 26. What is iron atomic 26? The what is the configuration? Argon 4s2 3d6. Okay, 4s2 3d6. In Fe plus 2, the loss of these two electrons, and that is convert the argon 4s0 3d6 state. Means contain the 6 electrons. That is both state, that is a plus 2 only. So here in deoxy form. So what is that? So that is acting as a means a high spin complex. That is acting as in what? High spin complex. High spin means electrons are means 6 electrons are there. No, these 6 electrons are arranged in the d orbital. In which form? High spin form means what? 5 d orbitals are there, 6 electrons are there. How to arrange the electron high spin means according to Hund's rule. According to Hund's rule. So what is the Hund's rule is given information what? Electrons are first single electrons are arranged. After that pairing is takes place like that here. Here pairing is there here. You observe their configuration deoxyform. Here pairing is there. Okay. Then here unpaired electrons are there. That is a high spin form. What is the form of that? High spin or outer orbital complex. We can explain in the last semester. Outer orbital complex, spin free complex, spin free complex. Okay. So what is the nature of this? That is can acting as a paramagnetism. What is the magnetism of that? Paramagnetism. Okay, paramagnetic in nature means what? Fe plus 2 is contain the 6 electrons. In that 6 electrons are arranged in the d orbital. Follow the Hund's rule. Follow the Hund's rule. Why? Because of that is the in here iron is acting as a high spin nature. High spin means outer orbital complex or spin free complex. Okay, that is a paramagnetic. Then in this high spin complex or deoxyhemoglobin form of the iron is bind with the oxygen and convert the oxyform bind with the oxygen in sixth coordination sphere. Okay, already fifth 5 coordinations are occupied, fifth is vacant, that is occupy the oxygen, then convert the sixth coordinate, then convert the sixth coordinate in that sixth coordinate form. In that sixth coordinate form, that is low spin complex. What is the spin of that? Low spin complex. So in that low spin complex means not follow the or not obey the Handstool. Same Fe plus 2 state is there. Okay, Fe plus 2 is there. That is two electrons are there, not five. So that is also Fe plus 2 is showing that Fe plus 2 state is there. Fe plus 2. Okay, in that state that is contain the six electrons. In that six electrons in low spin condition. What is the low spin condition? Electrons are arranged in not follow the Handstool. Electron arrangement means not follow the Handstool means how many electrons are there? Six electrons are there. The six electrons are arranged like this. In that oxyform is convert the diamagnetic. What is the form of that? Diamagnetic. Okay, so like that we can discuss the oxy and deoxy form of hemoglobin. Okay, then we can discuss about the what is the binding mode of O2. This is important. What is the binding mode? What is the binding mode of different binding modes of binding mode? Two types. That is called what? Geometry and term. What is that? That is called what? Geometry. Okay, so two scientists are explain the two types of binding mode. Two times scientists are explain the two types of binding mode. One is the Griffith binding mode. One is the what? Griffith, and one is the Pauling structure. One is the Griffith and one is the Pauling. Griffith structure. Pauling structure means what is the meaning of this? Means the how to bind the oxygen to the sixth coordination sphere. How to bind with here in Pauling structure. You observe the Pauling structure. Okay, in that Pauling structure is what ind on terminal. Okay, what is the binding mode of that? Ind on terminal. Okay, and that is a Pauling. Then what about the Griffith? That is the what? Side on terminal. What is that? Side on terminal like this. Side. Both are different binding mode is there. Side on terminal. Ind on terminal. Side on terminal. Okay. So which is a stable? The indon terminal is the stable form. Which indon terminal is a what? Stable form. This is a what? Unstable form. Means oxygen is bind with the unstable form. Why? Because of that is transport is takes place now. One place to another place. The sixth coordination space, this oxygen is replaced by another oxygen. Replaced by another oxygen. Okay, like that. So this is a suitable means binding mode of the binding with the oxygen in hemoglobin 4 ring, which is that is a Griffith structure or Griffith mode is the means like mode is what how to bind ఎట్లా బైండ్ అయింది ఆక్సిజన్ ఓకే ఆక్సిజన్ ఎట్లా బైండ్ అయింది అంటే ఇండాన్ టెర్మినల్ గా వచ్చింది ఇక్కడ సైడ్ ఆన్ టెర్మినల్ ఓకే ఇట్లా ప్యారలల్ గా మీన్స్ 4 రింగ్ కి ప్యారలల్ గా బైండ్ అయింది సిక్స్త్ కోఆర్డినేషన్ స్పియర్ అక్కడేమో బాండ్ యాంగిల్ ఇస్ దేర్ వాట్ ఈస్ ద బాండ్ యాంగిల్ ఇస్ ద ఐరన్ 120 degrees బాండ్ యాంగిల్ అక్కడ ఉంటుంది ఎక్కడ మీన్స్ పౌలింగ్ స్ట్రక్చర్ ఓకే దిస్ ఈస్ సమ్ వాట్ స్టేబుల్ ఇస్ దేర్ ఇది స్టేబుల్ గా ఉంటుంది ఈ ఫామ్ స్టేబుల్ మీన్స్ వాట్ ఈ నాట్ ఈజీలీ రిప్లేస్ ద ఆక్సిజన్ ఇక్కడ నుంచి ఆక్సిజన్ రిప్లేస్ అవ్వాలి కదా ఆక్సిజన్ రిప్లేస్ ఈజీగా అవ్వదు ఇక్కడ పౌలింగ్ ఫామ్ లో అదే గ్రిఫిత్ ఫామ్ లో అయితే ఈజీగా రిప్లేస్ అవుతుంది మనకి ఏంది రిప్లేస్ అయ్యేది కావాలి అంటే మీన్స్ టూ types of geometries are there means geometries are known as what binding mode okay so here 120 angle is there in the Pauling structure here 90 degrees okay what is that parallel means perpendicularly arranged here okay so like that we can discuss about the binding mode of oxygen it's a very important question for your examination point of here coming from for one question. Okay, these all are the what? Hemoglobin, binding mode of hemoglobin. Okay, like this. So this is a simple form of the hemoglobin structure. Okay, deoxyhemoglobin. This is a oxyhemoglobin. Okay, coordination number 4. This is a coordination number 5. Okay, the fifth one is weakly binded or water molecule is there. So and this is about the hemoglobin. Then what is the role of globin chain? Globin chain is there, no? What is the role of that? So the globin chain means what? That is contain the four firing ring and transport the oxygen from one place to another place of the various part of the various part of the living being. That is the role of globin chain. That is called one protein, one type of protein. And the another important question is there here, what? Oxygen absorption isotherm. Okay. And cooperativity. Oxygen absorption isotherm and cooperativity. Is the explain one scientist? Who is the scientist of that? Bohr. Bohr is the scientist that is based on that. This is Bohr effect and cheptam. Okay. Oxygen absorption isotherm is known as what? Bohr effect. What is that? Means how to saturate the oxygen in after absorb the oxygen to hemoglobin or any pigment. So what is that means oxygen absorption also means saturation is what is the saturation of that. Saturation means how much absorb. Okay, that is how to explain that. That is the partial pressure of muscles and partial pressure of lungs. Partial pressure of muscles and partial pressure of lungs. Okay, depend on this, we can explain the saturation of the oxygen. Okay, muscles అంటే మన మజిల్స్ ఉంటాయి కదా, ఆ మజిల్స్ లో ఉన్నటువంటి ప్రెజర్ ని బేస్ చేసుకుని యాజ్ వెల్ యాజ్ లంగ్స్ లో ఉన్న ప్రెజర్ ని బేస్ చేసుకుని ఆక్సిజన్ అబ్జార్ప్షన్ ఇస్ దేర్. సో జనరల్ వాట్ ఈస్ ద పార్షియల్ ప్రెజర్ ఆఫ్ ద మజిల్స్ అండ్ లంగ్స్? దట్ ఈస్ కాల్డ్ వాట్? బీపీ అంటారు, బ్లడ్ ప్రెషర్. Okay, how much that is? 120 by 80 systole and diastole. 120 by 80 is a common means partial pressure of the muscles and lungs. Okay, so based on that, what is how to explain that? How to explain that means what is the absorption means saturation and pressure, partial pressure. You take the one graph. Okay, flat the one graph. How to flat that? X-axis and y-axis we can take. X-axis and y-axis. In the y-axis we can means observe the saturation of oxygen. That is called SpO2. Saturation of saturation percentage of oxygen. That is called SpO2 antam. Then in the x-axis we can take in what the partial pressure of the muscles and partial pressure of the lungs. Partial pressure of the muscles and the partial pressure of the lungs. Okay, this is all the considered. Then pH of the condition also, then like that. So in the partial pressure is we can means BP. ఎంత చూడ how to consider that? 120 by 80 is the common. Sometimes low BP also is there, high BP also is there. That is the range is the up to can take the 60 to we can take the 160 140 up to 180 వరకు we can చూడొచ్చు మనం బీపీ అంటే 180 వరకు ఉన్న that is no problem, means saturation is there. Okay, this is in between the partial pressure of the lungs and muscles లో lungs లో partial pressure ని base చేసుకుని we can observe the okay means explain the saturation of the oxygen. So in this region in this region in this region almost the saturation of oxygen is the 100% is there, means 100% means 95 or 95 to 99% or 100% is there. ఈ పార్షియల్ ప్రెషర్ లో ఈ కండిషన్ లో ఓకే saturation of oxygen, okay, saturation of oxygen is 95 to 99% ఉంటుంది. That is compulsory. So below 90 is not considerable. Below 90 means that is we can suffering from different problems means okay like that. So then you can go to any hospital, you can means admit the hospital, they are arranged in one ECG machine. In that machine is you can observe three types of values are observed. వాట్ ఈస్ ద ఏం చెప్తాం మన ఈసీజీ మనకు కనెక్ట్ చేసినప్పుడు మనం హాస్పిటల్లో పోయినప్పుడు ఉన్నప్పుడు పోయినప్పుడు ఏమో మనకి వాట్ ఈస్ యూ కెన్ అబ్జర్వ్ వాట్ ఈస్ ద బీపీ. మనం ఏం చూస్తాం దానిపైన బీపీ కనిపిస్తుంది మనకు. ఓకే, పల్స్ రేట్ కనిపిస్తుంది. పల్స్ రేట్ దట్ ఈస్ మనస్ లైక్ పీక్స్ వస్తాయి కదా, పల్స్ రేట్ వీ కెన్ అబ్జర్వ్. అండ్ వీ కెన్ అబ్జర్వ్ వన్ ఆఫ్ ద కాలమ్ ఇస్ దేర్ దట్ ఈస్ SpO2. SpO2 also can observe. Okay, that is almost మనకు 96-97% కనిపిస్తుంది మనకు రీడింగ్ జనరల్ గా. ఓకే, 96, 97, 98 ఈ రేంజ్ లో ఉండాలి. బిలో 90 is not acceptable, means saturation is not there, means what our lungs problem is there, మన లంగ్స్ కెపాసిటీ ప్రాబ్లం ఉంటుంది కాబట్టి అది saturation is decrease. Okay, like that. Okay, based on that we can plot the one graph. All pH levels is the 100% saturation is there. Okay, 100% saturation is there in this region. What is the region? The blood pressure means partial pressure of lungs and partial pressure of the muscles. Okay, ఎంత 60 to 180 వరకు సాచురేషన్ ఎక్కువగా ఉంటుంది, మీన్స్ నార్మల్గా ఉంటుంది. ఓకే, 180 క్రాస్ అయిన బీపీ, 60 తగ్గిన బీపీ, దట్ ఈస్ నాట్ ఇన్ ప్రాపర్, means saturation is not takes place. Low BP is a very danger, high BP also is the very danger, means that is called what depend on our daily wise day to day activity. BP మన బీపీ పైన డిపెండ్ అయి ఉంటుంది. ఓకే, సో దిస్ ఇస్ ద రోల్ ఆఫ్ ద హిమోగ్లోబిన్ in living beings. Hemoglobin is one of the pigment that is contained in the different living beings. Those living beings are contain the red blood. RBCs are contain, that is contain the hemoglobin, that is contain the hemoglobin. So this is the Bohr effect antam. This is a compulsory exam lo adige question. What is that question? Explain or what, write a note on the oxygen absorption isotherm and the cooperativity. Cooperative means saturation. Okay, so then another pigment is there, that is what heme erythrin. What is another pigment? Heme erythrin. So we'll explain the ఇందాక చెప్పాను కదా heme erythrin, that is represented in HR. Hemoglobin represented HB. The hemoglobin is the heme erythrin. Hemoglobin is contain the iron plus 2. Heme erythrin also contain the iron, but two irons are present. Two irons are present. Okay, in the two irons is bind with the different types of proteins. Okay, or amino acids antam. Proteins are amino acid. This is the structure of heme erythrin. This is the structure of heme erythrin. So here two iron atoms are there. Okay, and in this two iron atom, this is the first iron, this is the second iron. Anukunnam. One is a two irons are there. First iron, second iron. In this first iron is what is the coordination number are there here? How many bonds are there? 1, 2, 3, 4, 5 bonds are there. How many bonds are there? 5 bonds are there. In the second iron, you can compare how many bonds are there? 6 bonds are there. 1, 2, 3, 4, 5, 6. In the first iron, 1, 2, 3, 4, 5. Only 5 bonds are there. So means this is the 5 coordinate, this is the 6 coordinate. So here deficiency is there. Okay. In this iron is two irons are surrounded by the or connect with the one of the hydroxy molecule, one of the hydroxy molecule, and glutamic acid and aspartic acid. Two acids are there here, glutamic acid and aspartic acid. These are the bind with the two irons, and remaining the histidine proteins are there. Histidine that is almost 120 or 140 histidine proteins are surrounded by the this. Okay, so many proteins are there. These are the considerable. their transfer the oxygen. This proteins. What is the almost 140 is there? Here numbers are there. So 54 histidine, histidine 25, histidine 72, histidine 78, histidine 101. Okay, like some numberings are there. So based on that, we can is a major role is there. That is globin chain like that proteins are this. Okay, so this is the structure of deoxy. What is that? Deoxy heme erythrin. What you find out here? One iron is a 6 coordinate, one iron is the 5 coordinate. So in this electron deficiency is there no? That is occupy the oxygen, occupy the oxygen and convert the 6 coordinate, convert the 6 coordinate. Okay, so then 5 is convert the 6, that is called what? Oxyhemoglobin, oxyheme erythrin. 5 is convert the 6, that is called what? Oxyheme erythrin. So after means bind with the sixth coordination of the oxygen, then here one type of bond is possible. Means in between the hydrogen and oxygen, in between the hydrogen and oxygen, okay, one bond is possible. What is that? Hydrogen bond. Intermolecular hydrogen bond is formed. Okay, like this. This is the oxyheme erythrin. After bind with the oxygen, so one of the iron is accept the oxygen from surrounding. Why? Because of in that iron is only 5 coordinate. The deficiency is there. Sorry, vacant orbital is there. In that vacant orbital is accept the oxygen from the atmosphere. Then convert the 6 coordinate. Then oxygen. After bind the oxygen, one of the oxygen is bind with the iron. Another oxygen is bind with the means hydrogen bond is formed. Here what side on terminal is, what is the end on terminal mode is bind with the which mode? End on terminal. End means one side is the end on terminal. One oxygen is bind with the iron, one is bind with the hydrogen of hydroxide. Okay, so this is easily replaced by another oxygen to solve. So this is the heme erythrin. Two irons are there. One iron is 6 coordinate, one iron is a 5 coordinate. So this is a pigment. This pigment is present in invertebrates. అకశేరు కాలు అంటాం, వెన్నెముక లేని జంతువు జీవులు అంటారు కదా. Invertebrates, in especially insects. What is that? Insects. ఇన్సెక్ట్స్ లో ఉంటాయి అంటే కీటకాలు అంటాం మనం. కీటకాలు insects lo the type of pigment is present. That's why in this insects are contain the blood is blue color. What is the color of blood? Is a blue color. Means grasshopper, మిడతలు చిన్న చిన్న పురుగులు ఉంటాయి చూడండి, పొలాలు పురుగులు, that is the blue color blood is present. So this is a heme erythrin. Then we can discuss about another. So this is a deoxyform, this is a oxyform. This is oxyheme erythrin. Oxyheme erythrin. Okay. So the remaining I will explain, all points are there here. This is the oxyform. Oxygen bind with the what here? You can observe here. Here bind oxygen. Where is oxygen bind? Sixth coordination sphere. Okay. Van der Waal forces means six coordinations is a intermolecular hydrogen bond is formed. Oxyform. This is a glutamic acid, aspartic acid, histidine, histidine. So many histidine proteins are there. Histidine. Okay, like that. So this is the heme erythrin. Then the next one is the hemocyanin. What is that? Hemocyanin. That is also one type of pigment. That is also oxygen carrier. Heme erythrin also, sorry, hemocyanin is also oxygen carrier. That is a pigment. That is a what? Pigment. Hemocyanin. That is you can represent what? HC తో రిప్రజెంట్ చేస్తాం hemocyanin. So that hemocyanin is contain the copper is the copper plus 1 is the metal is present, two coppers are there. How many coppers are there? Two coppers are there. So like this. This is the hemocyanin, structure of the hemocyanin. First you can observe. కనిపిస్తుంది కదా మీకు? Okay, this is the copper. This is the copper. In between the copper, one van der Waals force is there. Means attract both in weakly bonded van der Waals bond. Then the distance is the 3.5 angstrom. In this copper distance is there. How much length is there? 3.5 angstrom. Here almost 300 in the both coppers are surrounded by the almost 300 histidine proteins. Okay, in that some major proteins are surrounded or approach two copper. One side is in this copper is a 3, this copper also is histidine. Then their numberings are there, 314, 228, like different numbers are there, almost 300 histidine proteins are surrounded by that. So this is which form? Deoxy form. What is the form of this? Deoxyhemocyanin. Means here copper is the 4 coordinate sphere. What is the copper? 4 coordinate means 1, 2, 3. This bond means that is a weak bond is the now 3 coordinate. Then that is bind with the oxygen. How to bind? Side on terminal. What is the mode of that here? Side on terminal. This oxygen is a bind with the both coppers. Okay, like this. Here means what is there no energy means what the distance is there no in between two that. So side like that. This is the oxygen. This is the oxygen. This copper bind like this. Side on terminal. Then so this is convert the 3 to 4 coordinate. This is not 5 coordinate, 4 coordinate. This 4 coordinate. Okay. So the like that bond is formed. That is which form? That is the oxyhemocyanin. What is that? Oxyhemocyanin. Okay. This is the deoxyhemocyanin. This is the oxyhemocyanin. Okay. So this is also carrier the oxygen carrier. Okay. ఇది కూడా ఆక్సిజన్ ని ట్రాన్స్పోర్ట్ చేస్తుంది. So this is also one pigment. This is present in the which type of living beings? This is also Annelida type of family, the invertebrates. Okay, invertebrates, Annelida type of families. Arthropoda, Annelida, like that. Means cockroach is contain the blood, different color, brown color blood is there. Okay, phyla means that is also kind of white blood. White blood is contain the white color. White color is a deoxyform and brownish color. Brownish, white, brown mixed combination color is what? Oxyform. Okay, means phyla, cockroach means earthworm. Okay, like different types of living beings are there. That is their blood is a different color. Why the blood is different color? That is contain the one of the pigment is what? Hemocyanin. So hemocyanin is contain the what is the element? Copper. What is the element? Copper is the metal is present. Okay, like this you can discuss about the another pigment is what? Hemocyanin. Another pigment what? Hemocyanin. Like that you can discuss about. Three pigments are there in living beings. These three pigments are transport the oxygen throughout body of the part or throughout body parts. Okay. Then inhale is the oxygen, exhale is the what? Carbon dioxide. Who is it carry the carbon dioxide? So that another pigment that is the myoglobin in the mammals, different types of animals, living beings, human beings is contain the myoglobin also. That is the what? Exhale. That is carry the carbon dioxide from all body parts and remove outside. That is the role of myoglobin. Hemoglobin is there, myoglobin is there. Both are present in the contain in the iron is the metal. Okay, there is no iron, oxygen transfer is not possible. This is a very important role of the iron. Okay, that is we can calculate the hemoglobin percentage. Okay, different types of percentage are there. Okay, in childhood or means kids one type of percentage, adult one type of percentage, women's one type of percentage, means depend on the hemoglobin percentage we can means transfer the oxygen supply is there. So the deficiency of iron, what happened, that is a sickle cell anemia is the disease. What is that? Sickle cell anemia means nutrition deficient is there. So iron is generally deficiency is means what, nutrition we cannot follow the proper nutrition or proper diet, so iron deficiencies takes place. ఓకే, సో మీన్స్ దట్ ఈస్ కాల్డ్ సికిల్ సెల్ ఎనిమియా అంటాం మనం. ఏమంటాం దాన్ని మనం? సికిల్ సెల్ ఎనిమియా, న్యూట్రిషన్ డెఫిషియంట్ ఆఫ్ iron. Okay, so how to roll out that, we can means take the fresh vegetables, fruits, okay, means different types of what is dry fruits, okay, we can take. Sometimes we can use the iron tablets, or sometimes you can use the iron injections also available, or sometimes we can use the means incubate the blood also, transfer the blood translation also it takes place, means depend on the percentage of hemoglobin percentage ni base చేసుకొని మనం ఫస్ట్ ప్రికాషన్స్ తీసుకోవాలి. వాట్ ఈస్ దట్? డైట్, ప్రాపర్ డైట్ యూ కెన్ మెయింటైన్, యూ కెన్ మెయింటైన్ ద ప్రాపర్ డైట్, ప్రాపర్ టైం. ఓకే, సో ద హిమో మీన్స్ ఐరన్ ఈజ్ దట్ ఈస్ ఓకే, ఛాన్స్ ఎంత కావాలో అంత ఉంటుంది. దెన్ డెఫిషియన్సీ ఇస్ దేర్, వాట్ హ్యాపెన్, యూ కెన్ to approach the consult physician, then we can use the iron tablets, or depending on the hemoglobin percentage we can use the iron injections also, or we can use the sometimes iron means blood translation also can take place, like that. The very important role of iron, okay, in the hemoglobin, that is the oxygen carrier, oxygen transfer. So like that this also, what is that, in invertebrates, non-living, non means whatever, that is not అంత ఇంపార్టెంట్ ఏం కాదు మనం. So similarly, the another is there. What is that? Is that vitamin B6? In the vitamin complete organic substance, vitamin. This then here small topic is there. What is that? The very important question in the given examination point of view. What is that question? Compulsory question is given. What is the question of that? You can compare hemoglobin, heme erythrin, and hemocyanin. What is the difference between? Very important question. Very important question. What is the difference between hemoglobin, heme erythrin, and hemocyanin? What is the difference? Hemoglobin is present RBC. Heme erythrin is present the blue blood. Okay, hemocyanin is contain the white blood. Okay, hemoglobin is contain the iron. Heme erythrin also contain the iron, but two irons are there. Hemocyanin is contain the copper. Okay, like that. So many different points are there. Okay, so here so many different points are there. Some points there here, hemoglobin, heme erythrin, hemocyanin. Okay, so like we can explain that very important question. Okay, it's comparison, what is the difference between hemoglobin, heme erythrin, and hemocyanin? Hemoglobin, heme erythrin, and hemocyanin, what is the difference? Very important question this given for the examination point of view. Then after that we can discuss about the what, vitamin B6. Vitamin B6, that is we can give the name of that thiamine atom. Thiamine. So vitamin B6, the very important role is there in the living being. What is the role of that? That is, we can give the common name is the thiamine atom. Okay, that is three forms lo untai manaku based on their what formula, based on the formula of that. Okay, What is the formula of that thiamine? Okay, you can write the structure of vitamin B6. So what is this? This is a pyridine. Okay, so the heterocyclic compound. This is a heterocyclic compound. In this pyridine, we can give the numbering of the. This is the 1, 2, 3, 4, 5, 6 membered ring. This 6 positions are there. So in this pyridine, in the the third position is contain the alcoholic group CH2OH. Fourth also contain the alcoholic group CH2OH. And the fifth one is contain the hydroxy group. Sixth one is contain the methyl group. Okay. So this is the pyridoxine phosphate. What is the name of this? The chemical name is what? Pyridoxine phosphate. Pyridoxine. Pyridoxine phosphate. Okay, this is. And in the third position, so fourth position is bind with the replace the CH alcoholic group, replace the alcoholic group and present the CHO group. Aldehyde group is there. That is the pyridoxal phosphate. What is that? Pyridoxal. Okay, not phosphate. Later we can explain. Pyridoxal. First one, previous one is what? Pyridoxine. Means in the fourth position in this heterocyclic in the fourth position is bind with the pyridoxal. Another name also is there. Three types are there. Another type is what? That is contain the primary amine NH2. Okay, that is a pyridoxamine. Means three types of thiamines, vitamin B6, three forms are available. What is that? Based on the heterocyclic ring, pyridine ring is there. In that pyridine ring is contain the some different six positions are there. In different positions are present different functional group. Okay, in the third position alcoholic group, fourth position also alcoholic group ఉంటే ఫోర్త్ పొజిషన్ లో కూడా ఆల్కహాల్ ప్రైమరీ ఆల్కహాల్ ఉంటే దాన్ని ఏమంటాం మనం పైరిడాక్సిన్ అంటాం. అదే ఫోర్త్ ప్లేస్ లో సిహెచ్ఓ ఆల్డిహైడ్ గ్రూప్ ఉంటే దట్ ఈస్ కాల్డ్ పైరిడాక్సాల్ అంటాం. అదే ఫోర్త్ పొజిషన్ లో ప్రైమరీ అమైన్ ఉంటే దట్ ఈస్ కాల్డ్ pyridoxamine. Okay, three types are available. They are there here. You can observe. Just wait. This is also very important. Means this is involve the three types of reactions. So what is the reaction? I explain this. So this is what pyridine ring. So this is the first position, second, third position alcoholic group, fourth also alcoholic, fifth hydroxy group, sixth methyl group or alkyl group. That is the pyridoxine. Similarly, you can observe here pyridoxal, the pyridoxamine, three types of available. And so the which is the active, this is the active position. What is the active position? Like this. In this third position is there no? Third position ఉంది కదా. ఈ థర్డ్ పొజిషన్ లో ఉన్న ఆల్కహాల్ గ్రూప్ లో హైడ్రాక్సీ గ్రూప్ ఈస్ దేర్. ఇన్ దట్ హైడ్రాక్సీ గ్రూప్ ఈజ్ సబ్స్టిట్యూట్ విత్ ద ఫాస్ఫేట్ గ్రూప్. థర్డ్ పొజిషన్ లో ఉన్నటువంటి హైడ్రాక్సీ గ్రూప్ ఈజ్ సబ్స్టిట్యూటెడ్ విత్ ఫాస్ఫేట్ PO4 - 2 గ్రూప్ తో సబ్స్టిట్యూట్ అయితే దట్ ఈస్ కాల్డ్ ఫాస్ఫేట్ అంటాం. ఓకే, దట్ ఈస్ కాల్డ్ ఫాస్ఫేట్. ఇప్పుడు నౌ కెన్ బేస్డ్ ఆన్ ద థర్డ్ పొజిషన్ లో ఫాస్ఫేట్ ఉంటే దట్ ఈస్ పైరిడాక్సిన్ ఫాస్ఫేట్ అంటాం. పైరిడాక్సిన్ ఫాస్ఫేట్. ఓకే, సేమ్ థర్డ్ ప్లేస్ ఉంటే దట్ ఈస్ సిహెచ్ఓ ఉండి థర్డ్ ప్లేస్ లో ఫాస్ఫేట్ ఉంటే దట్ ఈస్ కాల్డ్ pyridoxal phosphate. Similarly, in the amine, third position is a phosphate, fourth position is a amine unte that is called pyridoxamine phosphate. That is called what? Pyridoxamine phosphate. These three forms are the active forms. These three forms are the active form. This completely in the organic substance. Carbon, hydrogen, nitrogen, oxygen తో ఉండి ఉంటుంది. That is formation of like this. That is called is a hydrocarbon, organic substance. Okay. So this vitamin B6 is involved in the three types of reactions. Three very very important reactions are there. What is that reactions? The first reaction is the transamination. Transamination means is involved in the amino acids. Alpha amino acid is convert the beta, amino acid is convert the ketone, ketone is convert the amino acids like that. Transamination, one of the important reaction. And the second reaction is the decarboxylation. Decarboxylation. And third reaction is the dealdolation. Dealdolation. These three types of reactions are participate in this vitamin B6. These three reactions are possible in presence of metal only. In presence of metal. What is our theme? Metal. There is no metal. These reactions are not possible. There is no metals are present. These reactions are not possible means what? The vitamin B6 is inactive. Vitamin B6 is there, but metals are not there. Reactions are these three types of reactions are not occur, are not do their metabolic system. Transamination is not occur, decarboxylation is not occur, and dealdolation is also not occur. So why? Because of there is no metals. There is the absence of metal. In presence of metal, these three reactions are possible. These three reactions are possible. Then we can explain their mechanism. Means decarboxylation, dealdolation, transamination reactions are means occur in the metabolic systems or digestion. Different systems are there. No, in the systems are these reactions are possible. So how to possible in presence of vitamin B6 as well as in presence of metal. There is no metal, this reaction is not possible. There is no metal, this reaction is not possible. Okay. So we can explain one mechanism. One reaction mechanism is there here. We can explain that. And so what is the role of that? Simple common reactions also is there. So the transamination reaction. You observe here, uh, transamination. Amine is transferred from one place to another place. So amino acid one here. What is the reactant? Common reaction. The amino acid one place react with the pyridoxal. Here convert the ketoamines to pyridoxal means alcoholic means pyridoxal means what? Aldehyde group carbonyl groups are present. Here convert that is what? Pyridoxamine. అంటే ఇక్కడ ఫోర్త్ పొజిషన్ ఇక్కడ ఆల్డిహైడ్ లో ఉన్న ప్లేస్ లో ఏమొస్తుంది అమైన్ వస్తుంది అంటే మీన్స్ ట్రాన్స్ఫర్ ద అమైన్ అమైన్ ఎక్కడి నుంచి వచ్చింది హియర్ అమైన్ ఇస్ దేర్ నో అమైనో యాసిడ్ అమైన్ ఇస్ దేర్ నో దిస్ అమైన్ is transfer pyridoxal to pyridoxamine. Convert the pyridoxal is converted into pyridoxamine. Okay. So like that pyridoxamine. Okay. Keto acid then convert the amino acid to and pyridoxal. Vice versa. This reaction is what? Reversible reaction. Pyridoxal is convert the pyridoxamine or pyridoxamine is convert the pyridoxal in presence of metal. In presence of metal. So what is here? We can what you have observed here? Amine group is transfer from one place to another place or one compound to another compound or one reactant to another reactant in presence of metal only. In presence of metal only. There is no metal. We cannot explain this here. Metal is there no here. There is no metal. Metal లేకపోతే this reaction is not possible. Reaction జరగదు. Okay, metal లేకపోతే metal ఉంటేనే జరుగుతుంది. So this is the mechanism of that. So the formation of the intermediate complex. So like that we can different types of mechanisms are there. These three mechanisms are similar. Means similar mechanisms are there. Transamination, aldolation and decarboxylation. These three types of reactions are same mechanism is there. In these three, one is given for the exam, one is given for the exam. So like that we can, these three types of mechanisms are there. Three types of mechanisms are there. Okay. So this is the role of vitamin B6. This is the role of vitamin B6. Then we can discuss another is there. What is that? Magnesium. What is the role of magnesium? Magnesium is present in the what? Uh, Chlorophyll. Magnesium is present in chlorophyll. So just wait. I will. So all of you follow the class. Just wait. I will share another. Uh, ఒక 1 minute ఆగండి, మళ్ళీ ఇంకొకటి షేర్ చేస్తాను. మయోగ్లోబిన్ మీన్స్ క్లోరోఫిల్ ఇది చెప్తాను. జస్ట్ వెయిట్. జస్ట్ వెయిట్. ఓకే. జస్ట్ వెయిట్. Next one is what? Magnesium. So what is the role of magnesium? Magnesium is present Mg plus 2 form in what is that? Chloroplast. What is that? Chloroplast. It is present in the plants. They are present in the plants. Okay, chloroplast. Chloroplast is contain the what? Chlorophyll. Chloroplast is contain the what? Chlorophyll. This is the structure of the chloroplast. So mitochondria, different types of is there. That is biology. We are not about that. Here what we can, chloroplast is contain the, chloroplast is a greenery part of the uh, plants. Okay, chloroplast that is contain the chlorophyll. Chlorophyll is contain the magnesium. Chlorophyll is contain the magnesium. So what is the role of chlorophyll? The role of chlorophyll is what? That is absorb the sunlight from the atmosphere. Sunlight, absorb the sunlight and do the photosynthesis reaction. There is no chlorophyll. Photosynthesis reactions are not possible, are not occur. Then whatever that food material is not prepared, not prepare the food material. Plants are not prepared the food material. Okay, so that is the role of chlorophyll. That is the role of, means magnesium is present in the chlorophyll. So Based on that, we can explain the structure of chlorophyll. Structure of chlorophyll. This is also similar. This is also similar what hemoglobin structure is there. Similar hemoglobin structure is there. But here we can consider that four five rings are there. We can give the numberings of that. We can give the numberings of that. This is the four five ring means pyrrole ring 1, this is a pyrrole ring 2, this is a pyrrole ring 3, this is a pyrrole ring 4. Four five ring and they are means like that. So in this two types of chlorophylls are there. Two types of chlorophyll. Chlorophylls are two types are there. One is the chlorophyll A and the chlorophyll B. Chlorophyll A and chlorophyll B. So how can you explain that? In the fourth pyrrole ring is there, no? In this fourth pyrrole ring, different pyrrole rings are contain the different different R. Okay, R means alkyl groups are contained. Just wait. So based on that, we can explain. R ni basis we can explain the chlorophyll structure. Okay, this is a observe here. This is a common structure for chlorophyll. The chlorophyll A or chlorophyll B. How can I explain that? This is the first pyrrole ring is contain R1, R2. So R1 say what is that alkyl group? Any one alkyl group is present. R1 that is the alkyl group. And second pyrrole ring also contain the R2, R3. And the fourth pyrrole ring is contain the R4. That is the alkyl group is substituted. So based on this alkyl R positions means R1, R2, R3 and R4 positions. Based on this alkyl groups, you can consider that a chlorophyll A and a chlorophyll B and as well as bacteriochlorophyll A and a bacteriochlorophyll B. Bacteriochlorophyll A and a bacteriochlorophyll B. Okay, means bacteriochlorophyll means that is green algae, fungus are there no. The algae, నాచు అంటారు చూడండి మన నీళ్ళలో ఉంటుంది. That is also do the photosynthesis. That is not present exact chlorophyll. That is a bacteriochlorophyll A. That is different types are there. We can explain. How to explain simply what is the structure of chlorophyll? So the chlorophyll means in center of the middle of the magnesium is means approach the four pyrrole rings. We can give the four ring numberings 1, 2, 3, 4. So the first pyrrole ring is contain the R1 alkyl group, and second pyrrole ring is contain the R2, R3, and fourth is contain the R4. The third is contain the one different group. I will explain that. What is the third? That is contain the cyclic. What is that? Carboxylic group is present. Cyclic carboxylic group is present. So cyclic ester group is present in the third. So these four are contain the four different. Okay. Based on that, we can explain chlorophyll A, chlorophyll B, chlorophyll means bacterial chlorophyll and the bacterial chlorophyll B. Okay. So like you can explain. What is the explanation here? You can observe here. You can observe this. What is that? Okay. So type R1, R2, R3, R4. So R1 means these are there no. R1, R2, R3, R1, R2, R3, and R4. So R1 is the means what is that ethylene group. R1 is ethylene. R1 place is present the ethylene group. R2 is present the alkyl group. R3 is present the alkyl group means saturated hydrocarbons. R4 is present the P. P means what? Pytol. Pytol. P is the pytol. G is the geranyl. Two groups are there. You can explain that. P means pytol antam. Pytol group. G means geranyl group. Geranyl group. Pytol group. R4 లో pytol group ఉంటే అంటే ఇవి ఉండి క్లోరోఫిల్ ఏ అంటే మీన్స్ వాట్ సేమ్ స్ట్రక్చర్ R1 ప్లేస్ లో ఇథిలిన్ గ్రూప్ R2 ప్లేస్ లో మిథైల్ గ్రూప్ అండ్ R3 ప్లేస్ లో ethyl group, and R4 is contain the pital, that is chlorophyll A. Then what about the chlorophyll B? Ethylene group, R1 position. R2 position is the carboxyl group, or sorry, aldehyde carbonyl group. Okay, and R3 position is the ethyl group, and R4 is present the pital, that is the chlorophyll B. Then what about the chlorophyll, bacterochlorophyll A? That is also in R1 position present the keto group, R2 present in the alkyl group, R3 present in the alkyl group, and R4 present in the pital or geranyl group. Pital or geranyl group. Any group is present, that is the what? Bacterochlorophyll A. Then what about the bacterochlorophyll B? That is in R1 position is the ketone group, R2 position is the alkyl group, and here R3 is the ethylene group is present. So here R3 ethylene group is present, and R4 is what? Pital group is present. That is the bacteriochlorophyll B. Means what is the difference? Chlorophyll A and chlorophyll B. Chlorophyll A is do the photosynthesis in presence of P680 nanometers range of the wavelength of that. That is P680. This is a chlorophyll A. Means six chlorophyll A is absorb the wavelength. What is the range of that? 680 nanometers. Chlorophyll B means what? P700. Okay. So chlorophyll B is absorb the wavelength, means sunlight. The wavelength is how much? P700 nanometers. So P680 is one type of plants are absorbed. P7 one type of plants are absorbed. This is the C3 plant అంటాం, దీన్ని మనం C4 plant అంటాం. Okay, Krebs cycle different is there in the your BSc intermediate or UG level. You can read about that. Means అక్కడ చదువుకొని వస్తారు. Photosynthesis means chlorophyll A, chlorophyll B, or photosynthesis PS680 or PS1 అంటాం. Photosynthesis 1 and photosynthesis 2, PS1 PS2 అలా మనం పేర్లు పెట్టి చెప్తాం. PS1 అని రాస్తాం రోమన్ లెటర్. PS2 means PS1 means 680 nanometers range, PS2 means 700 nanometers range wavelength absorb the chlorophyll, like that. Okay, so what is the difference? Small difference are there. What is the difference of that? Small difference means ఇక్కడ chlorophyll A కి B కి ఇక్కడ ఓన్లీ ఒక పొజిషన్ దగ్గర చేంజ్ ఉంది. వాట్ ఈస్ దట్? R2 or R3, ఎనీ వన్ పొజిషన్ ఇస్ చేంజ్. chlorophyll A R2 position, what? In the R2 position chlorophyll A is contain the alkyl group. In chlorophyll B is contain the what? Carbonyl group, not carboxyl, carbonyl group. Remaining are same. That is the only difference. So here what? P is the pital, G is the geranyl. Means in this P is the pital. 20 carbons are there. Alternative contain the alkyl group. This is the pital side chain. ఇక R4 ఉంది ఇక్కడ. R4 ప్లేస్ లో ఆ pital chain ఉంటుంది. R4 position is there no here. In that R4 position is the pital. In this position is the pital, P or G geranyl. Okay, like what is that? Like this. Geranyl means alternative double bonds are there. Alternative geranyl chain. This contain the 20 carbons. 20 carbons ఉంటాయి ఇక్కడ. All are saturated but contain the alkyl groups, but here geranyl is contain the alternative double bonds. Okay, these are involved in the photosynthesis reaction. Do the photosynthesis. Two types of photosynthesis reactions are there. Means the preparation of the food material, that is the two types are there in the form of energy storage, which form? ATP and NADPH2 form. What is the form of that? ATP and NADPH2. Okay, so two types of reactions are occur here. Two types of reactions means one prepare the food material in presence of the chlorophyll. In presence of chlorophyll, two types of reactions are occur. One is the cyclic photophosphorylation reaction and non-cyclic photophosphorylation reaction. Cyclic and non-cyclic. What is the cyclic? Cyclic means this is the cyclic. once start the reaction like this, once start the reaction like continue the reaction like this. Okay, so ఇట్లా స్టార్ట్ అయ్యి ఇట్లా పోయి ఇట్లా వస్తుంది, మళ్ళీ స్టార్ట్ అయ్యి ఇట్లా పోయి ఇట్లా వస్తుంది. దిస్ ఇస్ ద సైక్లిక్. వాట్ అబౌట్ ద నాన్ సైక్లిక్? స్టార్ట్ ద రియాక్షన్, ఎండ్ హియర్. నాన్ సైక్లిక్ ఇక్కడ స్టార్ట్ అవుతుంది, ఇలా వచ్చి ఇక్కడ ఎండ్ అవుతుంది. దిస్ ఇస్ ద నాన్ సైక్లిక్. ఓకే, దిస్ ఇస్ ద సైక్లిక్. ఇలా పోయి ఇలా వచ్చి మళ్ళీ ఇక్కడ వస్తుంది, మళ్ళీ ఇలా పోయి ఇలా వచ్చి ఇట్లా వస్తుంది, సైక్లిక్. సో ఇన్ దిస్ బోత్ రియాక్షన్స్ is the formation of what? Energy, food material. What is that? NADPH and ATP, adenosine triphosphate and nicotinamide, nicotinamide diphosphate hydrogen, nicotinamide, NADPH2, NADPH, nicotinamide diphosphate hydrogen. This is the form of the energy. Okay, ఈ ఫామ్ లో ఉంటుంది ఎనర్జీ అనేది. Okay, this is the cyclic and non-cyclic photophosphorylation reaction is occur. Do that reaction like this. Okay, so this is explanation. So you can I think this is the last session for inorganic chemistry. So I will cover minimum 70% syllabus is covered in offline and online mode. So remaining 30% is there. We can discuss about in our what is the practical session time. We can discuss. So I think this is shared in last week class. I will share this PDF. Okay, this PDF you can follow. You can means enough for your examination point of view. Okay, so this is the today class. Now stop the class. Then you can join the another class is there. Okay, thank you all of you.
సరే
Now I will tell me.
హలో సరే నేను
చెప్పండి.
నోట్స్ ఒకసారి మళ్ళీ షేర్ చేయరా సార్?
అన్ని మళ్ళీ షేర్ చేయాలా?
పీడిఎఫ్ అన్నీ ఒకసారి షేర్ చేయండి సార్. ఒకటే వచ్చింది సార్ అప్పుడు.
సరే, ఐ విల్ షేర్. నేను అన్ని ఆఫ్టర్ ఫినిష్ ఆఫ్లైన్ క్లాస్ అప్పుడే రెండు చెప్పాను, రెండు షేర్ చేశాను అప్పుడే. సో ఐ విల్ షేర్ చేస్తాను. వన్స్ అగైన్ ఐ విల్ షేర్ ద ఆల్ PDF. ఓకే
ఓకే సార్, ఓకే, థ్యాంక్ యూ.